Good morning, everybody. This is the video on sex-linked traits. This is our last type of inheritance that we'll be talking about and our last pattern of inheritance. And it is specifically about the traits, the genetic traits that are locked to the X and Y chromosomes. Typically, we'll be talking specifically about the X chromosome. So I do want to sort of preface this that I am trying to update this section as much as I can to better reflect the difference in terminology, and as the terminology is evolving on discussing sex, because sex and gender are two different concepts, as or at least that's how I believe in them. I follow that explanation, and also that the genetics that determine sex is actually much more complicated than we ever realize, and we discover new things about this all the time. About ways that the phenotype, the development of male and female sex organs, or male and female sex traits, those developments are actually highly influenced by several other factors, not just whether someone is born with a X or a Y chromosome or a pair of X chromosomes, etc. There are many other factors that simply just affect the phenotype, and then you compound that with the concept of gender, and this gets to be a complicated discussion point. But what we're really talking about here is an example where we can discuss the inheritance and how inheritance changes when it is on what is labeled as the X and Y chromosomes. So we have up till now only looked at traits that are inherited autosomally, traits carried on autosomes, where males and females are just as likely. Those that are assigned male or female at birth, those would be those individuals. All individuals are equally likely to receive that trait, because it does not matter about the there's an equal chance to inherit those traits. But in the case of of comparing an egg and a sperm, we know that the egg coming from a female reproductive system, it will always carry two X chromosomes. So this creates a difference, because the sperm can carry an X or a Y chromosome. And so this means that the statistics, the variation, the probabilities are different from the two different donors for any traits that are stored on those X and Y chromosomes. This all needs to probably change, but biological sex differentiation, this is an older terminology. We actually have moved away from calling it biological sex now. It is now known as assigned sex, is as I understand it, and this video will probably age the fastest out of any video I've made. Assigned sex, which is the idea that sex is often assigned at birth based on the phenotype, and that phenotype is often the result of the X and Y chromosome combination. So assigned sex differentiation in humans is typically the result of this X and Y chromosome. So all humans are diploid, and that means that they have 46 chromosomes on average. There are individuals born with trisomies or with monosomies, which means that they have only 45 or 47 chromosomes. But 46 chromosomes is the most common format. And of those 23 pairs of chromosome, one of each pair comes from the parent. During that fertilization, when these two haploid gametes meet, so these two gametes each carrying 23 chromosomes meet, the diploid chromosome number is restored in the zygote. So hopefully you remember that from the beginning of this unit where we discussed meiosis and mitosis. So genetically male and female, let's get rid of that too. So Phenotypically, you know, genetically. Well, yeah, okay. Without really exploring some of the more interesting cases for male and female determination, let's say genetically assigned male or female definition. So if an individual possesses two XX chromosomes, it will typically develop female traits. There are examples where this is not true, but such as various genetic changes that can happen. And if an individual possesses one X chromosome and one Y chromosome, then it develops male traits, typically. So this means that I'm going to just get my keyboard out. Oh, let's see what this looks like. Okay, I hope you didn't get too dizzy there. This means that something interesting happens where the female egg, this implies that all eggs coming from an XX donor will carry one X chromosome, and while sperm come from an XY individual, so they can equally carry an X or Y chromosome. In this way, it's actually the male sperm that determines whether a zygote will be carrying the XX or the XY chromosomes. And in that case, they determine the typical sex typical sex development of that zygote. So when Henry VIII had all of his wives beheaded because they could not produce him a male heir, it was actually his own fault, because it was his sperm that were stopping who were constantly donating X chromosomes. The X chromosome is essential to development. During the first 6 weeks, the embryo develops using information primarily on the X chromosome. So that is the case for a zygote that is XX or XY. Both of those zygotes will develop using important information on that X chromosome. And then during the 7th week, the genes on the Y chromosome will be triggered, and there'll be a release of male hormones, and those will start to develop to stimulate the development of the male phenotype. And those reproductive organs are actually made of the same genetic tissue, or the same tissue seeds, where the same seed cells are responsible for both reproductive organ developments, simply with minor changes, what we could probably call as epigenetic changes. So sex-linked genes are going to be those genes that are found, specifically found on the sex chromosomes, X and Y. These are the genes that will be altered by the combination of X and Y chromosomes that the individual carries. These genes, and their inheritance pattern is much more influenced, or is influenced, by the sex of the donor gamete. So, and the sex of the resulting offspring. So we can look over to fruit flies, for example. So eye color in fruit flies is a sex-linked trait, where red eyes are dominant to white eyes. So typically we would expect in a dominant recessive situation, we would expect that when we cross, say, two heterozygotes or whatever, we would get that same 3-to-1 ratio. But here, because the red eyes are attached to the X chromosome, they are going to have a difference in the inheritance. So if the recessive gene is R, a female carrying one copy of that gene, so a heterozygote, so she's still a heterozygote, will be crossed with a male. Now this male could be any of the chromosome, any type. If they could be white-eyed, like this male is here, or they could be red-eyed. And let's look at what that trait is going to do in a cross. So here's my Punnett square. I'm going to put that female on top, and I'm going to put the male to the right. So notice that the Y chromosome does not have that information on it. The X chromosome is more important to development, and so it's a larger chromosome typically, so it carries more information on it, and or at least it does in many animals. Plants are a little different. And so when we are considering these genes, we're almost always considering genes that are found exclusively on the X chromosome. So here we've produced another female who has red eyes because she is carrying that dominant gene. Over here, meanwhile, we're going to take that recessive gene from the male and the recessive gene from the female, and we produced a female with white eyes. Now, if we crossed the dominant gene here from the female with the Y chromosome, we have created a red-eyed male and a white-eyed male. And so in this way, we've created a very unique phenotype ratio that we've never seen before, which is one red-eyed female compared to one, so notice it's a 25, 25, 25, 25 ratio, 25%, one white female over to one red, and this is the male symbol, and I need space for one more, so I'm going to just shift that over to one white male. And so that is the case there. Another example of a sex-linked gene that we see in humans is hemophilia. This one is a very famous gene. In fact, it's one of the first genes that was ever identified in genetics. So hemophilia is a condition where blood clotting is inhibited due to the lack of the dominant gene responsible for making a clotting factor. So the dominant clotting gene, this is a capital H here, this gene is necessary to form a clotting factor protein. So without that gene, your blood cannot produce a certain clotting factor, and this makes the blood not able to clot, and that causes the recessive gene to become known as the hemophilia gene. It's a gene that's not effective. And so this gene is a problem because now the blood can't produce a certain clotting factor, and that can lead to the person bleeding out from even a small cut. And this is a very famous gene. It was known to be in the royal family, for example, the royals of UK, that is. And there's also been evidence of it in, I believe, one of the first geneticists in the Middle East from like the 600s, I believe. They had identified that hemophilia was inherited through family lines as well, and that was one of the first evidence that people had um, identified genetics as a methodology. So let's show an example. Let's do a carrier female. Carrier female crossed with a normal male. And so this means that we have an XH X carrier crossed with a male who is apparently normal in blood type. And the result will be that I have no space. Next year, rewrite these books with more space in them, in addition to changing some wording. Okay. Normal male. So in this case, we normally would expect that if you had essentially a homozygous male, right, and you crossed a homozygous male with a heterozygous female, you would expect all the children to be to have the dominant phenotype. But in this case, because the males actually only carry one copy of the gene, they only affect the females. Meanwhile, the mother is entirely in charge of the genotype of the males because they are inheriting those Y chromosomes from their dads. And here we end up with 25% affected, so 25% hemophilia, male. And the 75% are normal. And so that's where this sort of creates some interesting conditions. There are a couple of different types of color blindness that are, one of those types is sex-linked. The other one is not sex-linked, actually. And I believe recently I found out that my aunt and my dad both have the same type of color blindness. Or my dad has one type of color blindness and his sister has the other type. So anyways, okay, so color blindness, this is usually a color blindness genes result in a change in the formation of the color receptors in the eyes, causing difficulty in differentiating colors. There's a whole bunch of different examples of this. There are some fun, maybe I can find one real fast, but color blindness looks like. So yeah, so this is the image I was looking for right here. These are the 3 types of color blindness and what they would look like to you. And like if you were colorblind right now, then you could actually, if you're colorblind, you might look at this image and see 2 of them looking the same, the normal one and one of the 2 look the same. And I don't remember which one my dad is a carrier for, but that's okay, it doesn't matter. So in this case, I don't know why I used H in this example, but let's put that here. So we have a carrier woman, and we have a normal man, and so when they are crossed, we should have the same cross we just had. As I've said before, there are only so many different types of questions that we can really do in these genetics problems. I think that's why I'm really excited about doing lots of dihybrids this year. It makes a lot of different options there. So if red-green color blindness is a recessive sex-linked disorder, what is the chance that they will have children who are colorblind? And so there is the case. Okay, so I'm going to post some questions related to this type of of inheritance, and we'll take it from there. Hope that explains some things, and I look forward to talking to you soon.